Good afternoon, everyone. I'm Shile Qi from Brinatum Center, Institute of Automation, Chinese Academy of Sciences. Now I will introduce our work, MicroRNA132 associated multimodal neuroimaging pattern impaired in unmedicated major depressive disorder. Here is the outline of my talk. Firstly, the background. Major depressive disorder is characterized by affective, cognitive, neuromorphological, and molecular abnormalities that may have a neurodevelopmental origin. According to the World Health Organization, MDD will be the second leading cause of disability in the world by the year 2020, following the heart disease. Contrast to schizophrenia and autism disorder, depression is with relatively lower heritability, which has been estimated as 30% to 40%, while a growing number of studies have suggested that epigenetics plays an important role in MDD. Previous experimental evidence has demonstrated that MicroRNA132 as a brain-enriched microRNA plays key roles during both the early and late stages of adult neurogenesis, dendritic morphogenesis, and synaptic plasticity. A recent study demonstrated that serum levels of MIR132 were increased in MDD patients as compared with healthy controls, with a significant positive correlation between self-rating depression scale scores. Another study also demonstrated that MIR132 expression were increased in the peripheral blood samples of MDD compared to HC. In addition, in an animal model of chronic stress-induced depression, increased expression levels of MIR132 were detected in the hippocampus. Furthermore, several depression-associated genes, including MECP2, P250GAP, CREB, and parallel genes, were also experimentally validated as MIR132 targets. All above evidence suggests that MIR132 may have an important role in the pathogenesis and the neuronal mechanisms underlying the symptoms of depression. So, we hypothesize that MIR132 may be associated with alterations in two neuroimaging networks that are remarkably and consistently impaired in MDD, including the frontal limbic circuit and the hippocampus amygdala nuclei. Most studies about the role of MIR132 in depression are based on cultural neurons, post-mortem tissue analysis, animal models, or pure genetic comparisons. However, to date, no studies have directly linked MIR132 with multimodal brain imaging together in a joint analysis to investigate imaging epigenetic association and the underlying neuromechanisms of the depressive subjects. Each imaging technique provides a different view of brain function or structure. A key motivation for jointly analyzing multimodal fusion is to leverage the cross-information in the existing data, thereby revealing important relationships that cannot be detected by using a single modality. Now I will talk about the method used in our study. Here is the flowchart of the supervised three-way multimodal fusion method, and why we used MicroRNA132 as the reference to guide the multimodal fusion. Thus, enables extract the joint component that significantly correlated with MicroRNA132. Table 1 shows the demographic and clinical information of participants. Our study included 81 unmedicated MDDs and 123 age and gender-matched healthy controls. All these data were collected from the Mental Health Center of West China Hospital of Sichuan University. Here, unmedicated means to avoid the influence of medication treatment. We made sure that all of the MDD patients enrolled in our study had stopped taking any antidepressants for at least 3 months before scanning. Now I will introduce the results of our study. Firstly, the expression levels of MIR132 in whole blood. Since it is difficult to determine MIR132 levels directly from the brain of patients, increasing efforts have focused on measuring MIR132 levels in the blood. at the MIR132 levels in the brain of patients with mood disorders would be reflected by levels in the blood. We found that the expression levels of MIR132 in patients with depression were significantly higher than those in the healthy controls, which is in accordance with previous studies. The group difference between MDD and HC is 0.01. Secondly, MIR132 associated multimodal neuroimaging pattern. 3 imaging features, including 5 from resting state fMRI, FA from diffusion MRI, and gray matter from structural MRI, were used as multimodal features and were combined by supervised multimodal fusion, in which MIR132 was set as the reference. As in our results, one joint component was found to be group discriminating between patients and controls and significantly correlated with MIR132. As in our results, increased MIR132 levels in the blood of MDD were associated with lower 5 and gray matter values in the prefrontal cortex, hippocampus, and amygdala, with co-occurring lower FA in the forceps major and anterior thalamic radiation. Thirdly, the association with cognition and symptom. Correlation analysis between loadings and contact cognitive domains for each modality were also conducted. As in our results, higher expression levels of MIR132 were associated with lower RVP and IED cognitive scores. Here, RVP means attention test, and IED means executive function test. The joint component of DMRI also anti-correlated with the Hamilton Depression Rating Scale scores. For comparison, we also conducted the unsupervised method on the same dataset. It is clear that the unsupervised method cannot extract the joint component. Specifically, for SMRI, there is no component to be group discriminating. For fMRI, only prefrontal cortex was extracted, but without the limbic system, hippocampus and amygdala, as in our supervised results. Now I will talk about the main conclusions of our study. Fig A shows the mapping of 3 MRI components in the same brain with 3 colors. Note that the prefrontal cortex, hippocampus, and amygdala are all identified in both 5 and gray matter, constituting the frontal limbic system that has been implicated in mediating emotional processing and pathophysiology of MDD. These regions were also touched by the identified white matter tracts in green arrows. Well, this is all data-driven, further validate the effectiveness of multimodal fusion. Fig B shows the inference and summary based on our results. Higher MIR132 expression levels are related with lower 5 and gray matter values in frontal limbic circuits, result in poorer attention and executive functions in MDD. Note that the forceps minor interconnects the left and the right prefrontal cortex across hemisphere, and the anterior thalamic radiation links prefrontal cortex with thalamus. All this provides evidence for disrupted anatomical connections in frontal limbic circuits. Our results clearly support that key depression-associated brain regions previously implicated in both functional, anatomical, and structural brain studies may be affected by MIR132. In sum, using a data-driven supervised learning method, we revealed that MIR132 dysregulation in MDD may affect multifacets of brain function and structure on the frontal limbic circuit and hippocampus amygdala nuclei, which is the first step into determining the MIR132 variants on multimodal neuroimaging in unmedicated depression. These results implicate a possible brain network modulated by MIR132 levels in MDD, which awaits further verification and replication, and more research should be conducted in this field. Finally, I would like to thank the guidance and the massive effort from advisor Jing Sui and other colleagues and partners. Further information is available in our poster number 12 and 1215 and our related posters. Thank you for listening.